Greetings, everyone. Welcome to another edition of the Third Coast Water Seminars. My name is Elena Harkness. I'm the Executive Director of Current, Chicago's Water Innovation Hub. And on behalf of all of our presenting partners, Northwestern University, the University of Illinois Urbana-Champaign, Argonne National Laboratory, the University of Chicago, and the University of Illinois at Chicago, I'm thrilled to welcome you here for this really important conversation today with George Wells of Northwestern University. If you've just joined us for your very first Third Coast today, just a quick logistics items here. We do try to keep this really interactive, so please put Q&A, use the Q&A tab for your questions for George, and put any general comments in the chat tab. We'll take as many as we can, and really always love to hear what's on your mind. Also, this event is being recorded, and our recording link will be shared out. If you registered, you'll get that following the event. It'll also be posted to Current's YouTube channel, and those are fully public. You'll be able to see them and share them there. Our agenda is really straightforward. I'm going to be introducing one of our very close collaborators, Dr. Aaron Packman, who's the director of Northwestern Center for Water Research and a professor of civil and environmental engineering at Northwestern University. He is one of the co-founders and sort of masterminds behind the Third Coast Water Seminar Series, and he will be introducing our speaker. We'll have a 40-minute presentation, some Q&A, and we will get you out of here just close to the top of the hour. So that is our agenda. I'm going to give a little bit of an introduction to Current, why we're here. Current's Chicago's Water Innovation Hub. So we're a nonprofit organization that's 5 years young this year, and we have three main goals. Grow an inclusive blue economy, and by that we mean the people supplying and demanding water innovations and technologies, driving that innovation forward, and helping to solve persistent water problems here in our region and beyond. We're here because water is a global challenge, but it comes to ground here in our backyard. We're located here in Chicago and Illinois, and we feel today on a very heavy rain day, we all need less floodwater in our basements. We all need more clean and healthy water to drink. Those are common challenges shared and felt around the world. Water scarcity expected to affect 2/3 of the world's population by 2025. The climate shifts that we're feeling here today are affecting other places around the world, causing coastal erosion, flooding homes, polluted water to drink, and lots of other challenges. So these are things that we have to grapple with here in Chicago, but also around the world. And of course, water quality, thinking about emerging contaminants as well as known issues such as lead, getting those things out of our water infrastructure, and making sure we have clean sources of water to drink. So today's topic is going to tackle something that's really close to Current's heart, the nutrient challenge. We've been working on this here in Illinois for several years, and many other stakeholders for much longer than that. So just by way of quick framing, the excess nutrients, primarily nitrogen and phosphorus, that come from all sorts of things, from wastewater treatment to agricultural production, too much of that contributes to toxic algal blooms in lakes and rivers. Some of you may have heard of the hypoxic zone in the Gulf of Mexico, which is killing fish and marine life, bringing really expensive costs to the fishery and tourism industries. And of course, degradation to drinking water supplies, which has a huge price tag nationally. So we're really concerned about that as well. All in all, you can sum up these challenges as we need to rally faster to come up with innovative solutions to all of these different problems and help protect our health and the environment. And at Current, we believe that innovation takes partnerships, and that is why we are here with all of these collaborators today, these leading institutions that are driving forward water research across many different disciplines, many different kinds of technology, and we are here to sort of bring the best and brightest ideas all together in one place and try to move them forward faster together. We try to stay on top of the latest technologies, independently assess them, and then recommend them and help bring them to ground, sometimes with many of the partners here, to bring them into the real world and test them and try them out. Our evolving pilot portfolio, some of these topics we have covered right here in the Third Coast Water Innovation Series and others elsewhere, but we are working on sewage surveillance, PFAS elimination, finding ways to target and destroy forever chemicals, and of course, the lead detection and removal challenge, which affects many communities across Illinois and elsewhere. So that's just a broad sweep through what we're working on, but most importantly, we work in collaboration, and we are here to drive more partnerships to help solve water together. And the Third Coast Water Seminar is a really great example of that kind of collaboration and the power that this region can bring to bear on water problem solving. So with no further ado, I'm going to turn it over to, again, Dr. Aaron Packman, one of our closest collaborators and really one of the leaders and spearheads behind the Third Coast Water Innovation Series. He's going to introduce our speaker. So Aaron, to you.
Thank you very much, Elena. I'm very happy to have the opportunity to introduce one of my colleagues from Northwestern University, George Wells. George has a diverse background. He has undergraduate degrees in chemical and environmental engineering from Rice. He then went to Stanford University where he did a PhD spanning biotechnology and microbial ecology, and then went for a 2-year postdoc to Switzerland to EAVAG, which is the Swiss Federal Institute of Aquatic Science and Technology, one of the leading water research institutions in the world. George started as a professor at Northwestern in 2013, and his research programs rapidly expanded. He is one of our main points of contact for partnerships with wastewater utilities like our own Metropolitan Water Reclamation District of Greater Chicago, as well as other wastewater utilities around the US. He will address today some of the challenges with nutrients in wastewater, talk about opportunities for nutrient recovery, energy recovery, and other types of resource recovery. And with that, I will turn it over to George to start his presentation.
Thank you very much, Aaron. Thank you, Elena. Really a pleasure to be here. I've been a fan of Current's work for quite some time. Have interacted with folks from Current quite a bit over the years and really have enjoyed the seminar series. So it's really a pleasure to have the opportunity to contribute to it and to start a conversation around some of the work that we are doing, thinking about nutrients, this nutrient challenge, and in particular, thinking about coupling our goal of moving towards a water resource recovery approach to water management and nutrient management. How do we put these together? That's really what I want to focus on today. So the title of my talk is Towards a Circular Nitrogen Bioeconomy. I'm going to talk about some broad areas where we're focusing on advancing technologies and management paradigms for sustainable resource recovery, specifically nutrient recovery from wastewater. Just to make sure we're all on the same page, I'm going to zoom in on this phrase right here, resource recovery from wastewater, and talk a little bit about what this means and why we think this is important. My major area of focus is on engineered water systems, urban water systems in particular, specifically focusing on wastewater management. And resource recovery in this context is really a new paradigm in the way that we view and manage waste streams in general in society. Here's our current paradigm for, in general, for wastewater management in society. We take wastewater, it's high in oxygen depleting organics, high in nutrients like nitrogen and phosphorus, and other pollutants as well. In general, we send these to large centralized wastewater treatment plants that do a great job in general of removing these contaminants from water, but require quite a bit of energy and quite a bit of money. These are very costly, and they dissipate these contaminants usually to the atmospheric, carbon as carbon dioxide, and some as microbial biomass as well, waste microbial biomass, and then nitrogen to nitrogen gas in the atmosphere. And we produce clean water, of course, in these systems. For the most part, we dispose of that clean water in a receiving water body. This is a case of what I would call, and what has been termed in literature, misplaced resources. This organic carbon, these nutrients, nitrogen and phosphorus, the water in wastewater, these all have potential value. And so as we move towards a more sustainable management regime for water, it behooves us to think about ways to reroute these misplaced resources and close the engineered water cycle. Just a high-level schematic of what this looks like in my mind. We take used water. So I like to get rid of that word waste and replace it with used, which is very common in Singapore, for example. They talk about used water in Singapore. So we have used water that has organic carbon, has nutrients, has water, of course, in it. We send this to what's commonly called water resource recovery facilities. I like to think about these as microbial biorefineries, where we seek to extract products of value from this used water. We can think about things like bioenergy production, nutrient recovery, biomaterials generation, and moving forward, looking to the future, potentially platform biochemical generation as well. And of course, we produce clean water, and where it makes sense, we reroute that clean water for reuse, thereby closing the engineered water cycle. So this idea of resource recovery, promoting a circular urban water cycle is really key to what my team thinks about every day. As Aaron mentioned, I am an environmental microbiologist. So I work at the interface of really two fields, microbial ecology, where I like to ask questions about microbial community structure, function, dynamics, interactions, and biogeochemical cycling in both impacted and natural aquatic environments. But I'm also an engineer, so I like to ask questions about related to how we can put these microbial communities to work to perform useful services for society. For example, in wastewater treatment, bioprocesses that are extremely common to our urban water infrastructure nationwide. But most of the projects that we do in my group really lie at the interface between these two fields, between the fields of microbial ecology and environmental biotechnology, where we seek to deepen our understanding of the microbial world so as to develop new technologies, new management strategies for sustainable environmental protection, typically of the water environment, and public health protection, coupled to this newer goal of resource recovery from wastewater. This is really a wrapper, again, for pretty much everything we do in my team, something I'm really passionate about. We do have a specific focus in my group on tackling this nutrient challenge that Elena introduced. We're really interested in microbial nitrogen, which I'll call N, and phosphorus or P cycling, and ways of managing, removing nutrients from polluted water streams, but then also moving towards recovery of these nutrients as well. I'm going to focus primarily today on the nitrogen challenge, but I'm going to talk a little bit about phosphorus as well. So why are we interested in nitrogen? What is the big problem here? I think it helps to gain an understanding of big picture fluxes of nitrogen to the natural environment. So let's do that. Let's take a look at a plot of time on the x-axis, 150 or I guess 170 or so years now. On the x-axis and the y-axis, we're looking at, in blue, human population, and in pink here, we're looking at total anthropogenic reactive nitrogen inputs to the terrestrial environment. So I want you to note that both the blue line, human population, and the pink line here, total anthropogenic reactive nitrogen input, have increased quite dramatically, especially in the second half of the 20th century. And this sort of exponential increase in nitrogen input to the terrestrial environment really dates back, or the start of this increase really dates back to the invention of a very important industrial process, the Haber-Bosch process for synthetic nitrogen fixation. This is fertilizer production for the most part. Haber-Bosch process is extremely important to our industrialized food system in the world today. It is responsible for the fact that we're able to, for the most part, feed a burgeoning global population, but it has led to the alteration of the biogeochemical nitrogen cycle such that the rate of nitrogen has nitrogen input to the terrestrial nitrogen cycle has more than doubled from pre-industrial times. So we have this massive flux of anthropogenically influenced nitrogen. There's also phosphorus flowing into the environment. There are a number of negative consequences that stem from this dramatic increase in nitrogen flux. What are these consequences? Why are we interested in tackling this challenge of nitrogen removal and recovery? Ammonia is toxic to aquatic life. This is one of the key drivers of effluent limits from water reclamation plants or from wastewater treatment plants, removing that ammonia to protect aquatic life. There's also the related issue of high oxygen demand by ammonia, about 4.57 milligrams of oxygen per milligram of nitrogen. There's the issue of eutrophication or excess algal growth and resulting hypoxia or low dissolved oxygen zones in nitrogen limited systems. As Elena mentioned, right at the beginning of the seminar, the Gulf of Mexico is kind of the poster child for so-called dead zones, for hypoxic zones that arise seasonally due to nutrient pollution, but there are hundreds, probably thousands of these worldwide, and they're increasing in prevalence. So this is a major challenge going forward. There are also increasing concerns about emissions of the potent greenhouse gas nitrous oxide, or N2O, which is something that we'll come back to in a couple of minutes. And there are public health concerns, namely methemoglobinemia, which is caused by excess nitrates, NO3- or nitrite, NO2- in drinking water. This is particularly problematic in the Midwest here in the US, especially as you go inland from Lake Michigan, and cyanobacterial toxins that are produced by harmful algal blooms. And these are a major issue down the road in Lake Erie and in many of the lakes around our area as well. These are increasing in prevalence. So I think it's clear that this massive flux of nitrogen, of nutrients to the environment, is causing a number of very vexing problems, very vexing challenges. In fact, I'm going to highlight a publication that came out now 13 years ago from the National Academy of Engineering, highlighting 14 grand challenges facing the engineering community in the 21st century. And prominent among these is management of the nitrogen cycle. And if anything, this challenge has only become more severe in the last 13 years since the publication of this report from the National Academies. So what I want to do today for the rest of my presentation is I want to take a high-level view, start a discussion really about the challenge of nitrogen recovery from wastewater, what makes nitrogen recovery unique and particularly challenging compared to recovery of other resources like phosphorus or carbon from wastewater. And then I want to take a little bit of a deep dive into some of the work that we've done on one potential option for nitrogen recovery as bioenergy. We're going to focus on this molecule nitrous oxide, and I'm going to get into a little bit of the microbiology, the aquatic microbiology and bioprocess engineering that we do in my group. I won't go too into detail on the methods here, but I'll introduce this as a possible approach looking forward. And then at the very end, just for a couple of slides, I wanted to take a step back again, talk about a newer project that we're really just getting off the ground, and think about ways to potentially expand the product portfolio for nitrogen recovery from wastewater. So let's begin with this challenge of nitrogen recovery from wastewater. And to understand this challenge, I think it's helpful to understand our current linear once-through model for nitrogen management. This is essentially the route that nitrogen flows through society. Almost all the nitrogen that we use in society flows through this route. We start out with air, right? We're surrounded by nitrogen. Air is about 78% nitrogen. We use this industrial process that I mentioned, the Haber-Bosch process, to fix that nitrogen into ammonia. This is a very energy intensive process. Haber-Bosch is one of the major industrial processes, major chemical factories worldwide that are producing ammonia, thereby using about 1 to 2% of global energy use worldwide. What we do is we combine nitrogen from the air with hydrogen that is typically derived from natural gas. It's a fossil fuel intensive process under high temperature and pressure over a catalyst to produce ammonia. That ammonia is then the basis for fertilizer production, for industrial food production, and I want to point out for production of a wide range of high-value nitrogen-based commodity chemicals as well. So we use nitrogen in society, and for the most part, we have a once-through use of nitrogen. We also have a lot of waste, a lot of loss of nitrogen in these processes. That nitrogen is then lost in two major streams. One is to non-point source pollution, for example, agricultural runoff, major source of nutrient pollution that is extremely important, but it's not something that I'm going to tackle on this talk. The other major source of nitrogen pollution is point sources of nitrogen pollution, and the major point sources of nitrogen pollution are municipal and industrial wastewater. What do we do with municipal and industrial wastewater? If we're fortunate, if we're in a rich, wealthy country that has robust environmental controls, we may route that wastewater to a nitrogen removal process, a biological process for removing that nitrogen from wastewater, thereby producing clean water. And this is what we do in many parts of the US. Wastewater treatment on the whole, as I've mentioned, is very effective for many of its goals in terms of contaminant removal, but at present is a very energy intensive, very resource intensive endeavor. Wastewater treatment accounts for about 3% of US electricity use. And it turns out that about half of this energy use is estimated to be used to supply oxygen for the microbial bioprocesses that are really the cornerstone of wastewater treatment infrastructure. For nitrogen removal, we use a particular type of bioprocess, and I won't go into great detail about this. We call it the nitrification denitrification bioprocess. It's extremely common nationwide. There are thousands of full-scale nitrogen bioprocesses, nitrification denitrification bioprocesses that again work relatively well for removing nitrogen, reactive nitrogen from water, and protecting receiving water bodies. But they have a number of key deficiencies, including this energy demand that I just mentioned, very resource intensive, very energy intensive. They also produce relatively high levels of nitrous oxide, this potent greenhouse gas that we'll come back to in a couple of minutes. And in addition, and this is the key deficiency that I'm interested in addressing, or I guess one of the key deficiencies I'm interested in addressing, our conventional approaches to nitrogen management remove nitrogen from water, but they put it back in the atmosphere, right? So effectively what we're doing is we're dissipating reactive nitrogen that we've spent an enormous amount of energy fixing via the Haber-Bosch process. We're now spending an additional large amount of energy and large amount of money to put that nitrogen back into the atmosphere. It's a little bit counterintuitive. And the vision that drives a lot of what we're doing in my group in collaborations with Current and with utilities and with practitioners is to think about ways of kind of shortcutting this dissipation of nitrogen. Can we come up with economical and sustainable ways to move towards a circular nitrogen bioeconomy where we capture this reactive nitrogen in municipal and industrial wastewater and we reroute it for reuse? So designing a circular nitrogen bioeconomy for a future without waste or pollution. I just want to highlight a couple of new projects. This is something we've thought about for quite a number of years. There are a couple of new efforts where we're really expanding with a number of collaborators our investigation in this area. I'm going to mention two projects, both of which Current is very involved in. The first is the US-Israel Collaborative Water Energy Research Center, or COWORK. This is funded by BIRD. Aaron Packman is the US director of this center. And again, Current is a key collaborator, a key partner on this effort. And if you look at the bottom here of our organizational chart, we have a number of different technical projects. One of the core technical themes in this project is nutrients and energy recovery. So we're exploring with our collaborators across the US and in Israel a range of different approaches for both nitrogen and phosphorus recovery. In addition, in a separate but synergistic project funded via the NSF, we're looking at ways to move towards a circular nitrogen bioeconomy, really looking at innovations in process engineering via tandem bio and chemocatalysis for sustainable nitrogen recovery and nitrous oxide mitigation. And again, this is a collaborative project with a number of folks from Northwestern and from Stanford and an active industrial advisory board that includes Current. And so in both of these efforts and in a number of other efforts in my group, we're really delving into this idea of how do we actually promote recovery of nitrogen as a value-added product, a high-value product. And I want to point out that this is maybe a more challenging goal than it might seem. And I want to compare nitrogen recovery to phosphorus recovery and highlight some unique facets, some unique angles to nitrogen recovery. And by the way, we could also do this comparison to carbon recovery. A lot of the same distinctions for nitrogen hold true for carbon, a comparison to carbon recovery as well. The key point I want to make here is that phosphorus recovery, which is the other major macronutrient that we consider in water management, wastewater management. Phosphorus recovery from wastewater is increasingly common at full scale. There are many mature, commercially available phosphorus recovery technologies on the market. Ostara is perhaps the best known. In fact, the largest phosphorus recovery facility in the world is an Ostara process right here in our backyard at the MWRD Stickney Water Reclamation Plant. This is a process that produces a chemical precipitate, a phosphate precipitate called struvite. Struvite is magnesium ammonium phosphate. The facility at Stickney is capable of producing, I believe, around 9,000 tons of struvite per year, but there's a range of other approaches that involve similar chemical precipitation, production of chemical phosphorus precipitates that are on the market as well, most targeting struvite or brushite recovery. There is work to be done in phosphorus recovery. Our efficiencies aren't terrific. There's certainly some technology development and adoption that needs to occur, but this is a relatively mature angle for resource recovery. Similar to phosphorus recovery, there's been work for quite a number of years, and in particular over the last 10 or 20 years on lab scale approaches, really creative approaches to nitrogen recovery, largely from high strength wastewater, high-strength waste streams like anaerobic digester centrates or source-separated urine. Many interesting studies, many interesting papers focusing on a range of technologies, including ammonia stripping. There's been some great pilots and even full-scale testing of ammonia stripping from high-strength digester centrate, for example. In more recent years, there's been some great work on bioelectrochemical systems or electrochemically driven nitrogen recovery and other physicochemical processes like ion exchange and membrane processes. So there's been a lot of work in this area, but at present, in contrast to phosphorus recovery, nitrogen recovery, especially from mainstream municipal wastewater, which is the largest point source, the largest sink for waste nitrogen, remains extremely uncommon in practice. And I want to take a step back and ask, why is this? Why is nitrogen recovery unique? Why is it different than phosphorus recovery and carbon recovery from wastewater? I'm going to give you 4 reasons that I think play a major role in distinguishing our needs and our requirements, really, for nitrogen recovery. The first is that when we target recovery of nitrogen from municipal wastewater, we're dealing with extremely dilute feed streams. Mainstream municipal wastewater harbors nitrogen in the range of about 20 to 30 milligrams per liter of nitrogen, mainly as ammonia. And it turns out that our conventional microbial bioprocesses are very well adapted to working with these very low concentrations of nitrogen. It's very difficult to take many of these proposed nitrogen recovery technologies in the literature that have really been tested on high strength wastewater and apply these to this major source of nitrogen wastewater, municipal wastewater with these dilute nitrogen streams. They just don't turn out to be economically or energetically competitive with these conventional biological nutrient removal processes. So that's one reason that nitrogen recovery is so difficult. But for those of you who are familiar with urban water management, you'll know that phosphorus and carbon are also quite dilute in municipal wastewater. So this isn't really unique to nitrogen recovery. So there's a number of other factors that we have to consider. The second factor that I want to mention is that we have limited options at our disposal as engineers, as managers, for nitrogen concentration and recovery via what has been termed a partition, release, recover paradigm. A partition, release, recover paradigm. The basic idea here is that resource recovery is much more feasible in many cases if we work with a concentrated stream compared to a dilute stream. If we have a way to effectively and efficiently concentrate the resource that we're trying to recover to a side stream that's high concentration, we have many more options at our disposal for recovery. And a key example of this is phosphorus recovery. The way that we typically recover phosphorus in full-scale systems, there are some alternatives to this, but the basic approach, most common approach, is to use a microbial bioprocess called enhanced biological phosphorus removal. That's very common in practice for partitioning, for removing phosphorus from municipal wastewater. What we do in these processes is we select for a type of bacteria called polyphosphate accumulating organisms, or POWs, that are capable of essentially acting as phosphorus sponges. They take dilute phosphorus and they concentrate, they bioconcentrate that phosphorus. We can then separate out our microbial biomass, route it to a side stream. So we have partitioning and bioconcentration. We route that concentrated phosphorus to a side stream. We use a fermentation approach to release that into a liquid, a very concentrated liquid stream. Once we have that concentrated liquid side stream, chemical precipitation becomes relatively straightforward. Recovery is much, much easier in these concentrated streams. We don't have an analogous approach to bioconcentrate nitrogen as we do for phosphorus, and that really creates a challenge for nitrogen recovery. So that's reason number 2. Nitrogen recovery is so challenging. Reason number 3 that I think is really important to understanding economics of nitrogen recovery is that there are limited co-drivers for recovery of nitrogen compared to carbon and phosphorus. Let's look at phosphorus, for example. Phosphorus recovery has a number of major drivers for adoption. There are crucial co-benefits that come along with resource recovery with that production of struvite. A couple of these I've put on on screen. Struvite is often our target for phosphorus recovery, magnesium ammonium phosphate. We try to do this in a precipitation reactor, usually a fluidized bed reactor. It turns out that if we don't have that phosphorus recovery unit installed at a wastewater treatment plant, struvite in many cases will precipitate in any case, and it will precipitate in pipes or in pumps or in process equipment and cause major operational challenges. So it turns out that recovering the phosphorus also helps to prevent nuisance struvite precipitation. There are additional operational benefits of phosphorus recovery, including things like improvements in biosolids dewatering, and oftentimes limiting the need for very, very expensive mainstream capital improvements, for example, construction of new bioreactors or new bioprocesses for additional phosphorus removal. So all of these add together into major drivers or co-drivers for adoption of phosphorus recovery. We don't have these same types of co-drivers for nitrogen recovery. What that means is that the products that we target for nitrogen recovery have to be economically competitive on their own against our conventional nitrogen removal technologies. We can't rely on these co-benefits or co-drivers for adoption. And that major problem of products competing directly with our incumbent technologies is what brings me to my fourth point, which is that typically when you think about nitrogen recovery, our target for recovery is nitrogenous fertilizer production. That's a good target from a sustainability standpoint, from an economic standpoint. From a cost standpoint, it's really, really challenging. We're looking at the price of range of different nitrogenous and phosphorus fertilizers over about 15 years or so, years on the x-axis. And you can see, it depends on the type of fertilizer, but it hovers right around $500 per ton or so. It's a really hard price to compete with. It's very difficult, especially when looking at dilute, large volume wastewater streams, municipal wastewater, to compete with the current price of industrial fertilizer production. And so the key question that I want to ask, or at least I want to start a conversation about this, is how do we address these challenges? Are there other higher value targets for nitrogen recovery that can make nitrogen management and recovery more feasible, more economically feasible? And so I want to take this opportunity to switch gears a little bit and tell you about one option that we've explored for a number of years, just one option among many, and that is nitrogen-based bioenergy generation. We're going to switch gears and talk about microbial nitrous oxide production as a novel bioenergy source, really kind of a case study of one particular angle that we've explored in my group and are continuing to explore in a number of new projects as well. So here's the basic idea. What is nitrous oxide? Nitrous oxide is N2O. It's a very interesting molecule. It's a natural component of the atmosphere. In pre-industrial times, concentration of N2O is about 270 ppb, but it's increased quite significantly over the last 250 years. The current concentration in the atmosphere is about 330 ppb or so. And that's a problem because nitrous oxide is a very potent greenhouse gas. It's one of the two primary non-CO2 greenhouse gases. It has a global warming potential about 300 times that of carbon dioxide, and therefore accounts for about 6 to 7% of worldwide greenhouse gas emissions. Now, it turns out that most anthropogenically generated nitrous oxide emissions are actually microbially derived. They come from microbial activity in a range of different societal activities, including nitrogen management in wastewater. And I need to introduce a little bit of the type of microbial players, microbiology that we think about in my group to illustrate why and where this nitrous oxide comes from. So the microbial player that I want to introduce to you are so-called polyphosphate accumulating organisms, or POWs. I mentioned these previously as the key microbial functional guild for very common enhanced biological phosphorus removal processes that are used now at full scale for phosphorus management and phosphorus recovery from wastewater. These are the phosphorus biological sponges, if you will, that are commonly used in microbial bioprocesses. Most of these POWs are thought to affiliate with an as yet uncultivated group of bacteria termed Candidatus Acuminobacter phosphatus, when I include this phrase as yet uncultivated, that's kind of a fancy way that we use as microbiologists to say that we don't know how to grow these organisms in pure culture. We can't study them in pure culture in the laboratory. We have to study them as part of a community, for example, in a bioreactor. The way that we select for POWs is we actually use a bioreactor where we switch between different redox conditions. We start out with an anaerobic phase, so no oxygen. What we find is that these POWs, these bacteria, actually release phosphorus in the anaerobic phase. You can see phosphorus is being released as phosphate into solution here. If we follow that with an aerobic phase, then POWs then they reuptake this phosphorus into an intracellular polymer called polyphosphate here. Now, it turns out that some POWs are capable of phosphorus uptake not just under aerobic conditions, but also under denitrifying conditions. Denitrification is nitrate reduction to nitrogen gas. These are called denitrifying POWs or DPOWs. They're a very interesting connection between nitrogen cycling, nitrogen management, and phosphorus cycling and phosphorus management, but they're relatively poorly understood. In the context of this story, we're interested in DPOWs because it appears that DPOW-enriched wastewater treatment bioprocesses at full scale and in the lab appear to be particularly prone to generation of this greenhouse gas nitrous oxide. Where does this actually come from? If we look at our classical route for denitrification, this is the route that we use in thousands of denitrifying bioprocesses around the US, around the world. We reduce nitrate, NO3- to nitrogen gas. That removes it from water, puts it back into the atmosphere. But this denitrification, this reduction occurs through a number of different intermediates, including NO2-, that's nitrite, NO, nitric oxide, and here we are, N2O, nitrous oxide. And it turns out that under some conditions, this last step in denitrification can be inhibited, leading to accumulation of nitrous oxide. And of course, this is a potent greenhouse gas. In general, it's not something we want emissions of. And so there are extensive research efforts that seek to minimize its production in both engineered and impacted natural environments. And that's something that we're interested in. We're doing quite a bit of work in this area as well. But with collaborators, we've also realized that, yes, nitrous oxide is a potent greenhouse gas, but it's also a powerful potential bioenergy molecule. Nitrous oxide is used as a monopropellants in rocket engines. It's also used as an energy booster in race car injection engines. And so over the last several years with collaborators, we've used this idea of maximizing nitrous oxide production and capturing it as a bioenergy source to develop an integrated bioprocess that we call the coupled aerobic anoxic nitrous decomposition operation, which is a bit of a mouthful, but it comes up with this acronym, the CANDO process. We like very positive acronyms for our bioprocess names, where again, our goal is to remove nitrogen from wastewater, maximize its conversion by denitrifiers to nitrous oxide, but then capture all of that nitrous oxide, eliminate fugitive emissions, and use that nitrous oxide as a bioenergy source. In more recent years, the last couple of years, we've developed a twist on this CANDO process where we seek to combine nitrogen removal and bioenergy generation with biological phosphorus removal and recovery via selection for these so-called DPOWs, these very unique and poorly understood bacteria that tend to produce quite a bit of nitrous oxide. Here's just a picture of one of the systems that we've used for a number of years. We operated this lab scale sequencing batch reactor as a DPOW-enriched bioprocess for about 3 years. We fed this with synthetic wastewater. So we haven't tested this with real wastewater yet, but we have a new project that I think we'll have an opportunity to do that with a range of different types of real wastewater. But at this point, we've just used synthetic wastewater, targeting recovery of nitrogen as nitrous oxide as a bioenergy source. And here's a picture of if we zoom in on this reactor, the underlying microbial community. So just a little bit of evidence that we can actually do what we set out to do in this bioprocess. Remember, we're targeting phosphorus removal and recovery and nitrogen removal and recovery. Let's start out with phosphorus. We're going to look at just one cycle in this reactor. I could show you hundreds of these over the course of 3 years that basically all look fairly similar in terms of phosphorus and nitrogen recovery efficiencies. We're looking at just one of these. This is time on the x-axis. It's about 5 hours. In this reactor on the y-axis, we're looking at a range of different substrates and products. I'll draw your attention just to the orange line here, which is phosphates. And what you can see is that under the anaerobic phase of this bioprocess, we see a release of phosphate, exactly as we expect from a POW-enriched community. This is what we expect. If we follow this with an anoxic phase, so that redox cycling, we see then a reuptake of that phosphate. So we see quite robust phosphorus uptake under anoxic conditions. And when we couple this to a very short aerobic phase, we see very good total phosphorus removal, upwards of 90% in all of the cycles that we looked at of phosphorus removal and sequestration in microbial biomass. But what about nitrogen? Remember, our goal here is not just phosphorus removal, but also nitrogen management and recovery. What do we see on the nitrogen front? What we're feeding to this system is actually a nitrite-based synthetic wastewater, and I can explain in detail why we feed this in the questions section if there's time and if there's interest. Feeding nitrite to this system, what you can see is that we have complete removal of nitrite. This is just one reactor cycle, but it's very representative of what we saw throughout the entire course of the system. Near complete removal of nitrites in pink. What is that nitrite converted into? It's converted predominantly not to nitrogen gas, but to this green line here, to nitrous oxide. We see about 70 to 80% conversion in this mixed culture microbial bioprocess to nitrous oxide as a putative bioenergy molecule. And I want to highlight that this is an unusually high emission factor for nitrous oxide from a microbial bioprocess. In our typical nitrification denitrification processes, the ones that are very common at full scale, at Stickney, at O'Brien, at a number of the other water reclamation plants around Chicago and around the US, the typical emission factors are about 0.5 to 2% of influent nitrogen as nitrous oxide. So this is an unusually high percent conversion to nitrous oxide. We wanted to ask, why? What is the mechanism underlying these unusually high levels of nitrous oxide generation? And I'll stress that the answer to this question, I think, is important both for bioprocess development, for the CANDU plus P bioprocess development, but also more broadly for understanding and managing these unusually high levels of nitrous oxide production in systems where nitrous oxide isn't desirable, right? So in conventional processes, nitrification, denitrification processes and the like, we don't want fugitive nitrous oxide emissions. Understanding mechanisms underlying the generation of this nitrous oxide is really necessary to develop approaches to mitigate its emissions. So we evaluated a number of different hypotheses for why we were seeing this very high level of nitrous oxide production. I'm just going to take you through one of these that turned out to be quite interesting. The hypothesis here that we investigated was that perhaps in this bioprocess and maybe in nitrogen cycling bioprocesses more generally, we select for microbial community members that harbor incomplete denitrification pathways that lack genomic capacity for nitrous oxide reduction. Here's the basic idea. Microbial denitrifiers, I'm going to go a little bit into the microbiology here, but I'll try to walk you through it. Microbial denitrifiers are conventionally assumed to have an enzymatic pathway for complete denitrification. That means reduction of nitrates, NO3- in water, all the way to nitrogen gas, which is very poorly soluble, so essentially dissipates nitrogen to the atmosphere. That complete pathway includes 4 enzymatic steps. We have nitrate reductase, nitrite reductase, nitric oxide reductase, excuse me, and nitrous oxide reductase. So the core of our hypothesis is that perhaps in this bioprocess, we have selected for a community that contains community members that just lack this last step. They lack the capacity for nitrous oxide reduction. They lack this nitrous oxide reductase enzyme. Now remember, the key catalyst, the key biocatalyst in this and indeed in all enhanced biological phosphorus removal processes is Candidatus Acuminobacter phosphatus, which is as yet uncultivated. We can't just grow this organism in the lab and sequence its genome and evaluate the presence or absence or the levels of gene expression of these genes. Instead, what we have to do is we have to look at the entire community. And so the approach that we took to evaluate this hypothesis is we used a new relatively cutting edge approach to molecular microbial ecology. This is called genome resolved shotgun metagenomics. Basic idea is we take a sample of our reactor microbiome, extract the community DNA, and we just sequence all of that community DNA. Then we use bioinformatics tools to essentially put together the puzzle of genomes or putative genomes that are present in this community. And we end up with a range of different what are called metagenome assembled genomes, putative taxa that are present in this community. So I'm just going to take you through one of the core messages from this data, and then we'll zoom out and come out of the weeds a little bit. So what did we find in this CANDU plus P microbiome? We're looking at genomes. So these are bacterial taxa. On the x-axis, we have 39 different genome bins, high quality genome bins. On the y-axis, we're looking at just presence or absence of these core genes for denitrification, for reduction of nitrate to nitrogen gas. NOS-C is nitrous oxide reductase. That's the one we're most interested in. NOR-B is nitric oxide reductase. NIR-S and NIR-K are nitrite reductase, and NAR-G is nitrate reductase. And so the white boxes here indicate absence of a gene. The pink or red indicate presence of a gene. So just presence or absence of these key structural genes for denitrification. So a couple of key takeaways here: 39 different core high abundance genomes in this reactor. About 27 of them have at least one core denitrification gene. So a majority of these are putative denitrifiers. But of these 27 genomes, only one, number 14 here, harbored a complete denitrification pathway. And this was a surprise. What it showed to us is that incomplete denitrifiers or incomplete denitrifying pathways are surprisingly prevalent in this CANDU plus P microbiome. In addition, when we look at these 26 genomes of incomplete denitrifiers, what we find is that about 10, and these turn out to be relatively high abundance, genomes that increase in abundance over time in this system, completely lacked capacity for nitrous oxide reduction. They lack this gene nitrous oxide reductase. These are putative N2O producers that are very prevalent in the system. So coming out of the weeds a little bit here, we're excited about the CANDU plus B bioprocess as a potential route, one technology option, blue sky technology option for sure, but one technology option to rethink what is a resource and what is a waste product, rethinking nitrous oxide from waste to a resource as a means to recover bioenergy from waste. And also, I think what we've shown is that nitrous oxide production in this particular bioprocess and potentially in other nitrogen cycling bioprocesses is largely, or at least partially, driven by incomplete denitrifiers that actually lack genomic capacity for nitrous oxide reduction. We think this has important implications for understanding nitrous oxide emissions from both engineered and natural aquatic systems. So I want to switch gears just for a couple of slides. I'm looking at the time, and I'm going to go very quickly through this, but I think it's an important step to take important task to take a step back and think a bit more broadly with you about expanding the product portfolio for nitrogen recovery. We've talked about fertilizer production. We've talked about bioenergy production. Can we do better, and how do we evaluate various products that we could produce from nitrogen in wastewater? And this is really at the core of this new project that really just started a couple of months ago, collaborative project targeting various process engineering innovations for generation of high value products beyond conventional fertilizers from nitrogen. Key question I want to ask is, how do we identify those high value targets? What are the targets that are kind of low hanging fruit that we might be able to use to drive development and optimization and upscaling of technology innovations in practice? And I want to highlight a little bit of preliminary data from this project. Again, this is a very new project, but I'm really excited about it. Preliminary data from a very robust and very fun collaboration with one of my colleagues at Northwestern, Jennifer Dunn, who is a co-PI on this grant. Jennifer is a professor of chemical and biological engineering at Northwestern and is an expert in systems analysis. And so the approach that we've taken is to combine techno-economic and life cycle assessments with ongoing data collection from bench scale experiments and modeling of at-scale processes to evaluate what are these high-value targets that we might approach or we might use as key targets going forward for nitrogen recovery. And the approach or the results that we've obtained so far are really techno-economic in nature in evaluating bins of nitrogenous products. And I want to just mention two different broad bins that we're targeting for value-added product generation. The first are large volume but relatively low value products. Baseline fertilizer production falls into this category for sure. The market value for nitrogenous fertilizer is about $110 billion per year. The price, as I mentioned previously, is around $500 per ton. So that's our baseline comparison. But we're also thinking about generation of a range of other products, including one that appears based on our analyses to be quite promising, which is alternative protein generation. So this is animal or fish feed. The market value for animal and fish feed is about $251 billion per year. The price per metric ton isn't so high, but we think this is very promising because the potential processing or separations that would have to occur to produce this alternative protein is perhaps not so high, right? So production costs may not be so high for this product. In addition, there are potential co-benefits in terms of avoiding nitrous oxide emissions from common animal feed or fish feed generation via conventional industrialized agriculture. So we have this bin of high volume, lower value products that we're pursuing, but we also have a bin, and this is where I'm probably most excited, a bin of products that we have identified through our analyses that are much lower volume, but are much higher in value. And so I'll just mention a couple of them. One is synthetic hydrogels, for example, polyacrylates. This could be used, for example, as a slow release fertilizer in agriculture. And then we have a range of nitrogenous commodity chemicals, including nylon, which is commonly used in many different applications, engineering plastics and automobile manufacturing, et cetera. And then nitrile-based polymers like acrylonitrile and nitrile butadiene. And again, the market value, the total market value for these targets is quite a bit lower than alternative protein or than fertilizer generation, but the price per metric ton is much higher than we could achieve with conventional fertilizer production. And I think that's important, again, to rethink, to use these analyses, to rethink what are we actually targeting for nitrogen recovery in order to achieve a product that is cost competitive and energetically competitive in terms of the energy expenditure we need to put in to recover that product, to just removing that nitrogen from wastewater using conventional bioprocesses. So we're using a life cycle assessment and techno-economic analysis framework at this point to evaluate new technologies against alternative baselines for nitrogen recovery as value-added products, as animal feed, as fertilizers, as hydrogels, et cetera, compared to conventional generation from fossil fuels and conventional nitrogen management. And we're including in this comparison now, and this is work in progress, impact on not just product generation, but also nitrous oxide emission levels and rates of emissions. So with that, I think I need to wrap up. I'm out of time here, so I'll give you a couple of key takeaway points. The key takeaway point that I want to make, hopefully I've gotten this through, is that waste nitrogen in wastewater is not a waste product. It's a potential resource, but it's a unique resource. It's a resource that needs different consideration and different analyses and different approaches than phosphorus recovery or carbon recovery. And we need to make sure that we consider this in our search for technologies for approaches in terms of value proposition for this transition to a circular bioeconomy. One option that we talked about today is the CANDO plus P bioprocess for bioenergy recovery, but there's a range of other approaches. More broadly, hopefully I've given you some new understanding of the microbial world, which is where I love to work, that can lead to and convince you that we can use our new understanding of the microbial world to lead to sustainable bioprocesses for clean water, bioenergy production, and recovery of misplaced resources. With that, I'll thank my group and collaborators and funding sources, and hopefully we have time for some discussion and questions.
Great. I think we really appreciate the richness of that presentation. There's so much there. There's going to be way more questions than we have time for, but occasionally we'll do a little follow-up narrative Q&A, and we can send it to all the participants after. So that may be a good way to handle some of this. I am curious. So even with phosphorus removal, one of the big challenges is that I think, there's a couple of good examples of actually marketing that recaptured, recovered resource, but it's very challenging. It's not something that many utilities are equipped to do. So do you see pathways for recovered nitrogen to actually, translate from the wastewater setting into a marketplace? Like you've clearly mapped out, there's plenty of demand for these products, but,
Absolutely. Yeah, that's a great question. I think one of the deficiencies in resource recovery in general is that gap between the water sector and stakeholders, the end users, right? And we see this across the board in phosphorus and carbon, using biomethane, for example, and in nitrogen as well. And so that's one of the things that we're trying to tackle in some of these new projects. I think there's a lot more work to be done there. Mm-hmm. I will say one of the reasons I think that Ostara and some of the other phosphorus recovery approaches have been successful is that they take that challenge off the hands of utilities, and they take charge of the marketing and the sales of the product. And I think that's something that we're going to have to see in other resource recovery applications as well.
Yeah, it seems like there's some rich business opportunities there to kind of create that connection. So a couple of technical questions, if I can squeeze a few in. So one is, how are some of these, microbial biorefinery processes, how could they be complementary to non-biotechnologies? Like if you think about them relating to each other, I think you talked about some of that, but you obviously see other
right. Yeah, that's a great question, and that's really this new project that is just getting off the ground is really trying to tackle exactly that. How do we couple tandem physical, chemical, and biological processes to take advantage of each of these? So biology in general is very good at managing dilute streams of contaminants, and so we're interested in developing new routes for not dissipating nitrogen, but bioconcentrating nitrogen from wastewater, and that's completely new. I don't have any results on that yet, but hopefully in a little while we'll have some data and some results that we can show to you. But then coupling that to using the concentrated nitrogen for product generation, I think we need to turn to separations approaches and potentially to heterogeneous chemical catalysis, for example, for generating some of these products, right? And so combining these in tandem approaches, I think has a lot of promise going forward.
Mm-hmm. Okay. So there was a follow-up question on separation that I think we could handle offline. So back to the wastewater system, since that's where you focused. Is there an opportunity to do nitrogen recovery in a distributed way or closer to the source? Is there any sort of way to pair that with some of the more centralized approaches?
Great question. Probably at least on paper, if we were to redesign our water management system, the easiest way to recover nitrogen would be to install urine separation in every home and every office in every building. Because then you keep the nitrogen, you keep the nutrients separate, you don't dilute them to begin with. So absolutely, decentralized nitrogen recovery would really improve our options.
Right, and that would also, so home and business, but would that also apply in the agricultural setting?
So agriculture, there are two streams that I think are important to distinguish in agriculture. One is agricultural runoff, which is very, very challenging to think about from a recovery standpoint. I think, in my view, fertilizer, improved management regimes for fertilizer application, buffer strips for example, wetland restoration, and potentially development of slow release fertilizers to minimize runoff are probably more promising than direct recovery because of the distributed non-point source nature of agricultural runoff. But there are point sources of agricultural nitrogen pollution, for example, confined animal feeding facilities or livestock. So nitrogen recovery from manure, from concentrated animal waste is actually something we're quite interested in. I think there's quite a bit of promise there.
That was a planted question because we're doing so much work on the trading framework here in Illinois around nutrients. So yeah, just really, really exciting to think about the future there. So if you could stop sharing, George, I'm going to close this out. And if other George Brigandy, my colleague at Current, could launch the poll, we're going to end it there. I'm going to flip back. Hold on one second. Yep, just brief audience satisfaction poll. And hold on one moment. Sorry, click. Would like to encourage everyone to save the date for Chicago Water Week. We'll have a special edition of the Third Coast Water Seminars and many other events. So QR codes there if you want to join to be a partner. We are welcoming partner submissions through July 9th, so get those concepts in. And please save the date for our next Third Coast Water Seminar coming up at the end of July with Dr. Ashlyn Stillwell, our friend and colleague from the University of Illinois at Urbana-Champaign, so keeping with the Chicago and Illinois University theme for the summer here. She'll be talking about smart cities and the energy water nexus. So that is all the time we have today. Thank you so much, George. Thank you, Aaron. Thank you all of the other partners in the Third Coast Water Seminars for being here. Current's powered by these partnerships. We're so grateful to have yours. And if you have other ideas about how we can help drive water innovation forward, please reach out. We'd love to hear from you. So have a great rest of the afternoon, everybody. See you soon.
Thank you all. Bye.